discussion. Good luck.
Okay, thank you very much. My name is Hamida, and today I'm going to present my master's thesis on the title of Effect of Increasing Temperature on the Swimming Activity of the Sardine from Two Populations. First of all, I would like to introduce this as the part of the SardiTemp project, which aims to assess the effect of climate change on the small pelagic fish, on the dynamics and ecology of the European sardine. So, as the climate change is the global phenomena induced by human activities such as the burning of fossil fuels, industry development, it may lead to the increase of greenhouse gases, which may lead to the increase of temperature. And according to the projection made by the Intergovernmental Panel of Climate Change, the temperature is expected to increase up to 4 degrees centigrade. This may cause the significant effect on the population density, distribution, development and behavior of marine organisms, especially the small pelagic fish, such as the sardines, the anchovies, the herrings, etc. So these species play both economic and ecological important role. For example, in the food web, they connect both the lower trophic level and the upper trophic level. And economically, they contribute about the 30% of the total world fisheries, so they are very important. Also, these species are very sensitive to climate variability, so they are the ideal candidate for assessing the impact of climate change on behavior, such as the swimming activities, because swimming is very, very important for predator avoidance, habitat selection, foraging, recruitment, survival, and fitness. So we aimed to assess the impact of exposure to high temperature on the swimming activities of sardines, which were collected from two geographical regions, Olhão, which is in the southern of Portugal, where the fish experience a swimming temperature of 15 degrees centigrade, and the other ones were collected from the Gulf of Lion in the northwest Mediterranean, where the fish experience a swimming temperature of 16 degrees centigrade. So as we may see, these fish, they live under different thermal regimes, so they may have show different thermal tolerance. Also, this model species we use has a wide distribution in the North Atlantic, Mediterranean, and the Black Sea. Also, they are planktivorous species, and they are used as food for top predators. They are highly migratory species due to changes of the hydrographic conditions such as temperature, salinity. Biological factors include food and reproduction. And also they are sensitive to climate changes. So this highlights the need to address the impact of climate change on these species. So they were collected from the Olhão and the Gulf of the Lion, and they were transported to the aquaculture station, which is IMBRSea, where all the experiments were conducted. And at the station, they were randomly distributed in 9 tanks of 300 liters. And there they were acclimated for 1 month. Then the temperature of the tanks were adjusted to the targeted temperature, which is 10 degrees centigrade, 20 degrees, and 24 degrees centigrade with 3 replicates per treatment. And they were maintained for 1 month also. And the swimming activities were then recorded for 15 minutes by placing the video camera 1 meter above the tanks after the 10 minutes of the habituation period in order for the fish to adapt the condition. Then the video analysis was performed by using a computerized free software which is known as ToxTrack, whereby the average speed, total distance of the fish, and the exploration rate were calculated. And we decided to choose the period of 30 seconds for each randomly in order to have the consistent results. So after collecting the variables, we analyze our data by using the generalized mixed models to explore temperature effect on the average speed, on the exploration rate, and total distance traveled by the fish. And we entered treatment and period of the video as fixed effect, and tank was entered as a random effect to account for multiple measurement for the same tank. And all these analysis were performed by using R software. And our results were as follow. The average speed, the swimming activities was affected by temperature, but it was not statistically significant. As we can see here in my graphs, the Sardines from France show greater performance in speed with increased temperature. This can be due to muscle contraction and increased metabolic rates. Also, it can be due to the adaptation of the fish to high temperature, because in the Gulf of Olhão, they live under high temperature. Also, the Sardines from Olhão showed high distance at 20 degrees centigrade, but it decreased at 24 degrees centigrade. This is because it can be that the fish are already adapted to the cold condition. But in the total distance and the exploration rate, the Sardine from France, they showed low with high temperature, while the Sardines from Olhao showed similar exploration behavior across all temperature. This might reflect more pause event of fish from France, because in swimming, the pausing swimming can be used by the fish to stabilize the visual field, or it can be used by fish to allow more time for them to receive and process all relevant stimulus from the surroundings. So in these studies, there are some limitations which we found. For example, there were few replicates per treatment, and this is because some videos were of poor quality, so the software did not recognize. So we omitted almost four videos from both populations. So the sample size automatically became low. Also, there was high variability of the data, because some fish in the tanks behave differently from the same treatment. Others had higher speed than the other ones. So the conclusion, this study showed that the increased water temperature might have the potential to directly affect these species through changes in swimming activities. I think further long-term experiment should investigate the direct survival implication of the altered behavior found on these studies. And this will provide valuable information on this fish vulnerability to future global warming, and further contribute to awareness to decision makers and those who manage these natural resources in the face of climate change. I would like to thank my supervisor for her assistance and others who helped me during this study. Thank you very much.